So it's been said that we're 98% chimpanzee, and that's because if you take your 3 billion base pairs of DNA, or yours or anyone's, and a chimpanzee's 3 billion base pairs of DNA and line them up, they're going to be pretty much the same sequence of A's, T's, C's, and G's, give or take 2%. And once we have that genetic information, we can take it and wind back the clock to about 6 million years ago, when there was just one species of ape between the two. And half of them split off and became chimpanzees eventually, and half of them split off and led to us. But if you go a little bit farther forward, you can see that 1.5 million years ago, that group of chimpanzees was split in half by the Congo River. And on the north half of the Congo River, the north bank, they evolved into common chimpanzees, which we tend to think of as a little bit more aggressive. And on the south side of the Congo River, they evolved into bonobos, which are more like the fun-loving flower children of the primate world. And one of the great things about these processes in evolution is that the processes that happened in the past are the same as the processes that are happening today. And that means that evolution and speciation in primates is still going on. So if you go out to China, you can see that the Mekong River is splitting gibbon populations into two. For about sometime in the last 10,000 years, they've been starting to build up differences. And if you go to where I work, in the mountains of southern Yunnan province, you have what are called sky islands, which are these little rings of forest at very high altitudes. And below that, all of the land is deforested. So the gibbons in these sky islands can't cross between the different fragments. As a result of that, they tend to build up differences over time, and they start evolving and adapting locally. Now, these are all really interesting, big questions, at least I think so. But one of the difficulties is, how do you really answer them? How do you get this information? You need DNA, and you need a whole lot of it, right? And when you're working with species that are endangered or critically endangered, maybe there's only 1,000 of them left. What are you going to do, right? You can't shoot them, and you certainly can't go up with a cheek swab and send it off to 23andMe. So you have to find those non-invasive DNA samples that have the most lowly of origins, the fecal sample, because every time an animal defecates, a little bit of skin cells slough off from the intestinal linings, and we can extract DNA from there to answer our questions. But there's still one more catch, right? How do you find something greenish-brown on the forest floor that's brownish-green? Well, we're visually directed primates, right? But we can't actually see this very well. But we can use our big brains to find an animal that can smell it very well. I work with a dog, because a dog is a scat detection dog, can smell anything from bombs to drugs to bed bugs. And its nose is so powerful that it can smell 1 to 2 parts per trillion of airborne odorants, which is just tens of thousands of times what we can smell. A dog right now isn't just smelling the subway wafting in from the room, it's smelling the meat, cheese, bread, and whatever ranch dressing is made of. So sometimes a dog's nose can smell what a human can't see, and the biggest of questions have the smallest of answers.